over to Mark Field, who will be chairing this session. And each session has 30 minutes allocated time, 18 minutes for presentation and 12-minute discussion. And then subsequently, we'll converge together to have further discussion. Over to you, Mark, please.
Okay, thank you very much, thank God. And yes, welcome everybody to the Grand Challenges session. I believe what we're going to do here is going to be a 18-minute or about presentation from the Grand Challenges leaders for each of these topics, and then followed by 10 minutes of free-form Q&A. I'm going to make the executive decision of devolving the moderating of the Q&A to the leads, because I think that might be easier, because they know necessarily which questions that are coming up either in the chat or orally that they want to pick up and they think are the most appropriate. So if that's okay, that would be good. So I will certainly be prodding people after about 16 minutes to wrap up and move to the Q&A. So without more of ado, we have the basic biology question. The basic biology challenge, which is being led by Eric Linton, Michael Ginger, and Peter Myler, and focuses on the surface and mechanisms of gene expression. So would one of that trio please kick off? It looks like Peter. Oh, Eric.
Yeah.
Okay.
So I'm going to lead off on this, and we have our presentations together. So I'm going to do the lead off, but there'll be a bit of mixing in here with Peter and then Michael at the end to cover things. So our kind of challenge here was the basic biology of euglenoids, what we know and what we can do about them. So, sorry, I got to get on the right level. So the three challenges we were looking at were, can we harness the data from genome sequencing to develop robust forward and reverse genetics for one or more euglenoids, that is euglenoid genetics? Can we actually do some manipulation? From some of the talks we saw yesterday and today, looks like some people are possibly making inroads into that area. Two, evolution of the cell biology. How do euglenozoan cell shape and form and cytoskeleton work? Michael will be talking mostly about that area. I'll be talking mostly about challenge 1. And challenge 3, Peter will talk about, is more toward the reconstruction and evolutionary history of metabolism in euglenids, an evolutionary map of this and how the metabolic pathways have developed. Peter and I's positions will be kind of mixed together since they have a lot of relationships. And then the kind of the third part of the talk, or second part, depending on how I want to divide it up, will be Michael talking about the cell itself. So just a little bit of background from doing some looking around as of Genbank. And I'm sure there are more out there people have, but right now, from what I could find in Genbank, there are about 51 kinetoplastid genomes of various types, nuclear and mitochondrial. Many of these are multiple species of the same thing, so different culture collection versions of the same genus and species. There seem to be about around 20 euglenoid chloroplasts or plastid genomes available, probably a few more, but that's what shows up in Genbank. And only one euglenid. Right now, Euglena glacialis, the one from the paper with Thank God et al. And there's the nuclear and some mitochondrial genomes. I know there's been some more work done on that, but these are the ones that are kind of deposited and available for everybody to look at. Okay, and yeah, I don't know why it's being weird. So some of the challenges we thought to enable euglenoid genetics, we need better understanding of many things. First, the genome organization. We had a lot of talks on this. Not only the nuclear, but the mitochondrial and the chloroplast genomes and how these interplay with each other. Questions to ask are polycistronic transcriptomes or individual promoters. There's been work done in this area, but it's still not clear in many of these areas. Introns and exons, they do have them. The chloroplast has them. The nuclear genome has them. Mitochondrial maybe. 5' and 3' untranslated regions, signal sequences. Again, how are these genes controlled? Triploid, are these really triploid? You saw some talks yesterday, we're talking about are they haploid or diploid? We saw some evidence yesterday that they could be triploid. So what are they? This will come back to this in a few minutes as well. Regulation, still working on that. Transcription, messenger RNAs, translocations, pre or post translational controls. What are they? Again, evidence for a little bit on both sides, but a lot of mechanisms still need to be worked out. And again, how universal are these mechanisms? Or is it going to be one set of controls or mechanisms and, euglena or euglenoids, photosynthetic, the non-photosynthetic, the kinetoplastids compared to the other ones? Do we have a mixture? Again, many of these factors are unknown. We know that in Leishmania, we can carry that over and see if those apply in Euglena itself. DNA repair, again, some of this is from Peter. Homologous recombinations, how long is the template do we need? Or non-homologous enjoining. So again, how is the DNA repaired? How is the DNA maintained in the cells? RNA machinery, again, the machinery appears to be present, but does it work as expected? Is it one mechanism? Is it the only mechanism? Again, these are a lot of unknowns still. And then gene families. Are they functionally redundant? Are the genes appear in families? Is there redundancy between them? Can you knock out one? The origins of these families. I'll pause for a second if Peter wants to say anything in addition to this slide.
I'd like to put a little context in here. I guess gene regulation, I think, it's important to know what, if there is regulation at the level of transcription, that's going to be important. We heard talks yesterday indicating that it's mostly post-translational. That's going to provide potentially some issues in terms of understanding how to regulate your genes that you want to do in forward genetics. And in terms of the DNA repair, that was really about, well, okay, how do we get CRISPR-Cas to work? Will it work in trips where you can have short, like in trips where you can have short template regions to get homologous recombination and knock out or pop in your gene of interest? If it doesn't work that way, it's going to be trickier.
Continue on. So this is what things that we need to actually do genetics and manipulate euglenoids at some point. If we're going to continue evolutionary studies, we have to have better understanding of the origins of the genomes. Again, transfer of genes from the nucleus, is it universal, is it variable? Which genes have moved from the chloroplast or mitochondria into the nucleus? Is that variable or not between the different groups of photosynthetic for chloroplast or across all the euglenoids? Do they have the same, was it one giant movement or not? Do all the pathways have a common origin, or is the nucleus a mosaic? Again, there's evidence for biochemical pathways being assembled from different sources. The slides. Tetrapolin synthesis pathway and Calvin Benson. These are just a couple of papers from Rich Tremer, who was my PI when I was getting my doctorate. So I'm most aware of his work on this area because it came out of our chloroplast genome work. But it does show that a lot of the pathways are mosaics of different sources, glaucophytes, red algae. So how widespread is this amongst euglenoids? And I heard a few people speak of this yesterday as well. Evidence of sexuality in this. And again, do they have sex? And I don't think anyone's ever seen it. We tried for years and couldn't make them have sex, or at least ever see anything that looked like sex. What's evidence? Is there some way to sort this out? Can we look for meiotic genes? The chance of doing that. Also, again, leading to this, are they haploid, diploid, triploid? Is that variable? How universal? Do we have some haploids, some diploids, and some triploids? So again, there's many things we don't know about how the structure is, and I see a lot of people are working on that. Economic uses, we heard a lot of that today. Again, bioremediation, medicines, food sources, and toxins. Again, manipulation and better understanding could help us use it better for all these different areas. Again, toxins can be used for medicines, but they could also be dangers we have to deal with using them as food products at some point, depending on the species we use. This next part here is Peter again. Possible solutions to some of these questions we have is, again, more euglenoid. We need more genomes. Again, that's the whole point of this. We need nuclear genomes and mitochondrial genomes. But again, which species? And we had a lot of candidate species, so this may be settled at least for the short term as to what we need to look at. Better assemblies. We need long read. The genome seems to be very complex in putting it together. Long read sequencing and high C sequencing seem to help a lot. A lot of people have shown that and discussed that yesterday, and I'm sure I'll get to discuss more in the evolution studies coming up. We also need better genome annotations. Again, RNA-seq, proteomics. Many people yesterday were talking about running certain things that you normally use. RNA-seq may not be the best case, so we may have to try different techniques or even develop some new techniques in order to understand how the gene regulation works and which genes are actually done in euglenoids. And I think so. So I don't know if Peter wants to say anything else before Michael comes over, and I'll drive, and then Michael, you can just tell me when to hit the button.
No, I think you did a great job there, Eric. We'll just wait for some more questions. We've already got some comments that we can discuss at the end.
Okay.
Okay, thanks, Peter. Thanks, Eric. Thanks to thank God and to the organisers for giving the opportunity to think around grand challenges. You're not really going to see the surface mentioned in the last 3 or 4 slides, perhaps because none of us was a particularly surface biologists, and we took the eye example of a surface as something we might choose to think about. Actually, we thought about some other things. And for Grand Challenge 2, we thought to simply ask, why do the different euglenids look the way they do? The image that you can see on the left-hand side of the slide is taken from an essay by Brian Leander and colleagues from a few years ago. It shows nicely the variations in shape, size and pellicle patterning that can be seen across the Euglenida. We can also ask how cells divide and how has that evolved or changed across the different lineages. And if we look within the euglenids, we can see that, as with many other microbial groups, there's a flirtation with multicellularity that we can also probe. And on the next slide, please, Eric, you can see that genomes, transcriptomes, they're going to allow us to predict, or on a basis of size, disorder, gene dosage, domain architecture, perhaps estimate cytoskeletal protein content and inventories. But if robust reverse genetics becomes available for multiple species, then we can begin to localize proteins or test gene function. And we can start to have a proper comparative evolutionary cell biology comparison with the kinetoplastids and perhaps comparison with the trypanosomatids and some of their adaptations to parasitism. 1 or 2 people have spoken about the advances in transgenics that are coming with Euglena gracilis. But perhaps one thing to remember is that here we might be looking at many examples of essential genes. And in order to have something that comes out the other end of a genetic manipulation, we've got to have a cell to look at. So a starting point where we can take a cell population, a stably transformed cell population, and induce the protein depletion by switching on, whether it be RNAi or a conditional gene knockout. Or if we go the transient way, the confidence that we hit all cells equally with the constructs that we're using to try and silence gene expression. On the next slide, please, Eric. Then if we want to think about that sort of wider academic comparison in terms of cytoskeleton evolution across the Euglenozoa and start to look at make comparison with cytoskeletal evolution in the kinetoplastids, then perhaps the key message to think about is that where work has been done, then often it appears to be that relatively small changes in gene expression or co-option of co-option of proteins into novel contexts often underpins quite large changes in cell morphology or cytoskeletal function. And what you can see on the left-hand side with the cartoons and the scanning electron micrographs is that we're talking here about the changes that see a transition from a phagotrophic biflagellate free-living kinetoplastid to a uniflagellate osmotrophic non-predatory parasitic trypanosomatid, where we have a microtubule corset that is tightly defining cell shape, and what you are, whether it's trypomastigote, epimastigote, promastigote, amastigote, well, that's defined by the position of your mitochondrial genome or kinetoplast relative to the nucleus and to the posterior cell end. That mitochondrial genome, it also moves from being in the vicinity of the flagellar basal bodies in the kinetoplastid example that you can see here. So the mitochondrial genome is still in the mitochondrion, but it's in the vicinity of the extra mitochondrial flagellar basal bodies. But by the time we've moved to the trypanosomatids, that mitochondrial genome is now physically attached through the mitochondrial membranes to those flagellar basal bodies. And on the next slide, please, Eric, you can see indication, just summary, of some of the proteins, including some conserved proteins that we've worked on with others that are essential for some of the cytoskeletal adjustments that have occurred during the early stages of trypanosomatid evolution and was summarized as I walked through the cartoons. We've also got examples of lineage-specific proteins such as the calpain GM6 or FLAM3, where the reduced expression of these proteins is sufficient to drive a shift from a tripo to an epi or a pro-mastigo-like appearance. And if we move from the static sort of or the picture of what is gene function, what is the contribution that a protein makes to cytoskeletal organization, then other groups have been probing in recent years the molecular mechanisms that underpin organelle duplication, segregation, or cytoskeletal rearrangement during cell division within the trypanosomatids. And within that work, often showing that familiar players from yeast or mammalian cell biology function in new contexts. So with the last slide then, please, Eric. Then we're moving to a challenge that very much speaks to the challenges around evolution, ecology, the environment, translational biology, and it's the reconstruction of inventories and thus histories for metabolism and sensory perception by euglenids. It's useful in many contexts to reconstruct those metabolic histories, perhaps in spotting the ways to make defined media for some taxa, or for spotting opportunities for metabolic engineering and synthetic biology. But we can also ask around gene families, gene clustering, the subtelomeres, lateral gene transfer. How do these phenomena contribute to the evolution of central and secondary metabolism in euglenids? There's rich literature out there showing how these phenomena contribute to metabolic evolution in other microbial eukaryotes and their near relations. We know only a little about the situation within the euglenids. Eric's touched on it a few minutes ago in his talk, and you can see on the left-hand side an image that was first shown by Mark yesterday. It's a phylogeny for nucleotidal cyclases, so the enzymes responsible for synthesis of key second messengers, cyclic AMP, cyclic GMP. And what you can see within the phylogeny in green, as Mark pointed out, is the big expansion of some cyclase families in Euglena gracilis. But the other key bit of information to note is that if we look within the domain architecture of those cyclases, then we can see that domains are present within some of those Euglena proteins, either responsible for detecting changes to light or contain the domains that are thought, such as HAMP domains, that are thought to mediate the conformational change that changes protein activity in response to external cues, such as changes in oxygen tension, the availability of other possible electron acceptors or other chemicals. And finally, it comes back to that first grand challenge and the development of robust reverse genetics for multiple euglenids that is going to allow us to most readily test gene function, be it in testing the functionality within euglenids of the enzymes, the regulatory proteins, or the structural scaffolds that make the cells what they are. And I think we are going to stop there, please, Mark.
Oops, yeah, thank you very much, Michael. And I think bang on time. I didn't even have to agitate at all. So great, thanks. Great presentation, covering a lot of ground, of course. So do we have questions, please? Either via the chat or over Zoom, please.
So Mark, let me start off by asking some of the genomics folks, and perhaps yourself, what is the evidence for or against polycistronic transcription? Is the current genome good enough to really be able to tell that? Or have you looked harder than I have? Because I couldn't tell.
I think I would say that I can't tell either. I mean, thank God should also probably comment here, because, you know, what I'm quoting really is his work. But we did find evidence of contigs with more than one gene and potentially RNA that could be mapped, spanning both predicted open reading frames. But of course, the problem there is that what I will be unsure as to whether what we're dealing with is just run-on transcription or whether it is true generation of polysystronic transcripts. So, thank God. Would you comment?
You probably mentioned it. It wasn't so clear from the work basically. So, and whereby we have multiple, maybe a single particular gene family, kind of having multiple representative. So I think maybe by the time we have maybe a complete data set, I think that would basically help us to determine that, because from the genome too, it wasn't that clear. And as you heard yesterday, it's also very fragmented, and also the intricacies that put as well.
Most of the coding sequence is, of course, transpliced, and it does have an exon which would tend to point towards polysystronic transcription, of course. But never say never as to whether all of them are, some of them are, or none of them. I've got nothing. I'd be grateful if anybody. Sorry, I had a comment here from Tony Russell. Is that it occurs at least for non-coding RNA classes. So, Tony, do you mean by this by sort of small tRNAs? Any more detail?
I think he can feel free to unmute if he needs to, since we have more time.
Or maybe he's.
Then someone is. Okay.
Okay. So, can I invite any more questions? Oh, sorry. Tony's just sent back, but it's to everybody. So these are small nuclear RNAs for sure.
Yeah. Someone is asking: is there now a consensus about Euglena gracilis not being haploid? So I think from the conversation so far, it's not clear.
Yeah, I think there's going to be a question of what do you mean by triploid or haploid or diploid. So until we actually assemble full chromosomes, that answer is not really going to be clear, and that is an enormous challenge at the moment. So we shall wait and see. Yes. I mean, they seem to have the content would indicate from what Neil presented yesterday that they're at least triploid, but that doesn't mean that they are they have trisomy in that they have 3 copies of each chromosome. They may have fused some chromosomes, and we don't know.
This is Eric. Does anybody have any feelings? I myself personally have always had a feeling that euglenoids, based on what we keep finding, that the nuclear genome, we're going to find it to be, I would say, more mosaic than we would expect for other ones, just because they seem to eat a lot of things and picked up different things at different points in time in different genes. Do we feel I think we're going to have like a core set of genes that are the same for kinetoplastids, etc. But I feel it's like a core set with a whole bunch of extra baggage tacked onto it. Like, oh, this piece is from here, and it picked up a piece, it kept it, and then it integrated it with other bits. I mean, just the few papers I have read, and again, my area is more of systematics and evolutionary relationships and not much in pathways, but from the few I've been involved with and looked at it, it looks like that way. Other people get that feeling, or am I just kind of really reaching out and going, it's kind of out there?
The analysis that we did, and really was mainly the work of Steve Kelly, in fact, who contributed a lot towards putting the Euglena genome together, to whether it's a genome or inverted commas genome, but at least set of contigs. He did quite a bit of that analysis, and very much we felt that what we were getting was consistent with Chris Howe's shopping bag hypothesis, that there were a fair number of genes that we could pick up that we thought were bacterial origin. They were not coming in from archaea, they were true bacteria, and they were of a fairly disparate origin. So it was unlikely that they had simply come in along with the endosymbiont that was producing the chloroplast or the mitochondria, but in fact we got multiple rounds potentially, and I think that's quite common for things with secondary plastics. I did have a comment down here from Michael Hammond, actually, we'll discuss this in the next session on evolution, so perhaps we should...
I really like that the shopping bag thing. When I heard that, I went, "Oh my God, that makes so much sense."
Yeah, very smart inside. Yeah, absolutely. Are there any more questions? Because of course we can move on if there are questions or comments. Question: will long-read sequencer solve the problem of the fragmented genome data?
So, I mean, I think Neil presented pretty good discussion of that yesterday. In my opinion, yeah, long-read sequencing with high C, we'll be able to get a pretty good assembly. I think that's clear. I mean, Neil knows more about it than I do, but I think that's going to be the solution.
Yeah. I agree. I think it's the only